CRISPR-Cas, which like scissors, biological scissors. We will tell specific cutting place, and they will cut only mutated, cancer-mutated genes. We will forget big surgeries. Genes and stem cells and AI will be drive whole of healthcare in the future.
In 2013, Hollywood actress Angelina Jolie shocked the world with her open confession. She announced that she had undergone surgery to remove both of her breasts. This bold decision was based on the results of genetic testing. The test showed that Jolie had a mutation in one of her genes, which greatly increased her risk of developing breast and ovarian cancer. The operation was successful and her risk of breast cancer dropped from 87% to less than 5%. After that, the number of women getting genetic tests for gene mutations sharply increased around the world. This phenomenon even became known as the Angelina effect. The story of Angelina Jolie showed the world that early DNA-based diagnosis can prevent tragedy. Awareness of genetic risks can save many lives. Kazakhstani scientist Amankeldi Salybekov is creating opportunities for the early detection of cancer, cardiovascular diseases, and rare genetic disorders. In his laboratory, modern methods of reading DNA using artificial intelligence are being developed. These technologies make it possible to detect dangerous changes long before the first symptoms appear. At the same time, Dr. Salybekov's team is working on gene editing technologies that allow for the precise removal of damaged DNA segments, reducing the risk of disease development at an early stage.
3 or 5 years ago, Chinese scientists already edited genes which carrying like interleukin-7. They removed those genes which is responsible for HIV, susceptible gene. They removed those genes and newborn baby without those genes. It's like modified human being. We are using gene editing tools, CRISPR-Cas9, which like scissors, biological scissors. We will tell and program with these scissors and tell specific cutting place, and they will cut only mutated, cancer-mutated genes. Genes and stem cells and AI will be drive whole of healthcare in the future. These 3 topics is the main topics in the healthcare. So, if you remember, in 20 or 30 years ago, we had like surgeries, this kind of like big surgeries, right? They are removing some such kind of organs. But nowadays, we have minimal invasive surgeries, like putting some kind of stents. They are replacing. But nowadays, I believe that, so we might have stem cells and gene therapy, which also will remove these stents, and we will forget these big surgeries.
A drop of blood, saliva, or hair can reveal the secrets of a person's health. Modern technology makes it possible to analyze these samples to understand which diseases someone may be predisposed to. Amankeldi Salybekov and his team are creating Kazakhstan's first genetic risk database. It will collect data on mutations specific to the country's population. This will help identify common genetic risks, develop more precise preventive measures, and improve diagnostics.
This is the samples from the patient's biospecimen, serum plasma. We are dividing into 2 tubes. We isolated using centrifuges, and one of the major point of this techniques is we want to extract or isolate circulating free tumor DNA. So we have here isolation kit with magnetic beads As you can see, here is the magnetic beads, which is binding with circulating tumor DNAs. Here we have serum, blood, two types of. So we can just select here as a serum because our sample is a serum, and then we can go to the next step. First is like binding, which is binding here with specifically solutions they have: lysis, washing, 2 steps, and elution, and the beads with the final. And finally, this automatic robot. 16 samples can be set in these parts, and 16 samples also can be set here. Overall, it can be isolated 32 samples altogether. This is automatic suction pipettes. Now you can see. Now is mixing all of the beads and trying to going to the next step is the lysis, and then the binding, and the lysis, and the washing step, and then finally isolating high purity genomic DNAs. Here we have several analyzing assays. First one is DNA, RNA. Also we can analyze the proteins, but in our case, so we will analyze the DNA. And here also we use this one and close samples and run the samples with the nanogram microliters. We'll go to the next machine, which call it the QSEP1. In QSEP1, so we will also check quality, RNA integrity scores, number, so ring numbers, which is important before going to the next step for library preparation.
Next, the scientist checks the purity and concentration of the extracted DNA using specialized equipment. This step allows the samples to be prepared for accurate reading of the genetic code.
We have 2 cartridges for library. This cartridge we will insert to here in this section. Special, they have the place A and B. We have to insert to here. Also, we have a small cartridge which we have to also insert to here. As shown here, we have 2 chips, and with 4 lanes. Here we are using FCH chip with high throughput. In one chip can be sequenced up to 80 whole exome sequencing samples. After completion sequencing, we will have reports. So which reports will be transferred to the server.
After that, we got FastQC file. It's a format of file like docs, Excel, and etc. And after that, we are analyzing of this file. In this file, we had information about DNA, about genome of a human. We use a pipeline which include data processing steps like trimming, alignment. We are cleaning our data. After that, we use variant calling and annotation. After annotation, we can saw is it variant, some of part in genome is pathogenic. If we will talk about statistics, 85% of all of mutations are located in exome. It's a protein coding part of genome of human. And we can, with this pipeline, with data analysis, we can identify these genes, these mutations, and we can early diagnosis part of disease, and in this case.
Amankeldi Salybekov grew up in a family that always valued the pursuit of knowledge. After graduating from university and completing his internship in Kazakhstan, he went to Japan to study molecular biology and genetics. In Japan, Amankeldi became the director of the Kidney Transplantation and Treatment Center at one of the country's leading hospitals. This experience allowed him to gain a deeper understanding of how new technologies can extend patients' lives. Soon after, he returned to Kazakhstan and together with a team of like-minded people founded the Kazakh Institute of Innovative Medicine.
My father was in veterinary scientist. He established a laboratory in village, especially. So in Soviet Union, to establishing laboratory in the village, it was so difficult. So he spent a lot of energy for that establishing laboratory. My elder brothers told me that some of the researchers from Science Petersburg and from Moscow, they came to this village and to gain some kind of methods, because it was like unique methods. So it was for me like inspiration, and as well as, so that is the meaning. It doesn't matter where you are working, where it's like location, it doesn't matter. So you can establish institute and working. When I worked in the hospital, so we had a lot of patients who is suffering from leg ischemia. So ischemia is the meaning of insufficiency of blood supply to the lower extremity. So you might know nowadays in Kazakhstan we have more than 1,100,000 patients which is suffering from this disease. It's a terrible pain, and we don't have cure for that one in Kazakhstan. So in that case, just I searched from the internet, and I was interested that in Japan they're developing new know-how technology for the stem cell therapy, which they can produce new vessels. They can create new vessels and transplant to the leg, and they can just save lower limb. So I was really interested. My curiosity, it's day by day it's increased, and that's why I just found this Asahara Synthesis Lab and moved to bring this technology to the Kazakhstan. And I spent over maybe 10 years for that.
Amankeldi Salybekov tries to spend his free time with his family. Together they play football and take short trips to historical sites. For Dr. Salybekov, it's an opportunity to share the history of their homeland and important events of the past with his children. Horseback riding also holds a special place in his life, a passion he developed in childhood. He believes that staying healthy doesn't always require going to the gym. Simple workouts at home and spending more time outdoors are enough to stay fit and active.
I try to not spend a lot of time to watching the movie, because it's really eating my golden time, and the social network as well. I like to discuss with a person who achieved for something. For example, so far I met with 4 Nobel laureates in medicine and chemistry and biology. So just meeting with them and with great scientists, just talking with them like face to face. So this is like making me happiness and encouraging to myself to go forward and learn from something from their achievements. By 2030, so economics are expecting that AI will be dominant In worldwide, it will be around 30% of whole GDP of the world, 30%. But second place will be biotechnology, including genetics and new treatment drugs. So we have to get our niche from AI and biotechnology. So we have to start from now if we will planning to get some kind of revenue, if we will try to make our life better in the future. So we have to start from now.
Time is an irreplaceable resource. For a scientist, it is twice as valuable, says Dr. Salybekov. The success of an experiment, the timeliness of a discovery, and most importantly, the lives of patients depend on how efficiently every minute is managed. Research cannot afford delays. Every breakthrough requires not only knowledge, but also precise planning.
We have to segregate, for example, essential things from the secondary things. So first of all, we have to define what we have to do in short-term, mid-term, and long-term period. So usually we are dividing these three terms, and we are defining our priorities. So essential, mandatory things. So we have to do. And the secondary and so forth. So for that one, Japanese methods for us is most important, and we are actively using for every time management, especially in our lab. For teamworks, also it's important. So if you remember, in most of post-Soviet Union countries, so we have some kind of culture. For example, one scientist should lead for everything, and around this scientist, all of the scientists should work and contribute for his or her, scientific purpose. In our case, so we have some kind of flexibility in our laboratory. So we are finding essential priorities in our team, and then we are just dividing depending on the ability or like some person can do in a short term, but some person can't do in, for example, even if you are giving like long-term period, so she or he can't complete. So we are defining and giving them flexibility to improve or self-increasing, like self-learning and finding solution by themselves. So this kind of techniques we are using.
Amankeldi Salybekov believes that the future of medicine is closely tied to the development of bioinformatics. This field brings together artificial intelligence, big data analysis, and multi-omics. The scientist has set a goal to establish the first faculty of bioinformatics in Kazakhstan. In his view, this will make it possible to train specialists capable of conducting cutting-edge research in multi-omics and bringing innovation into healthcare.
A lot of mutations don't, let's say, wake up unless there is some sort of environmental factor from the outside, maybe something they eat, maybe some other lifestyle choices they do. So that's why our research is very important in covering those multi-directional risks, let's say, to the health of individuals. If you look up any type of disease in Kazakhstan, most of the data on it is just statistical. So we know how many people are sick with it, maybe what areas of country are maybe more prevalent, but we don't know what specifically is happening inside those people. So we have to, study it, and there are very few labs that do that in Kazakhstan.
Amankeldi Salybekov is a face of Kazakhstani science. He explores the secrets of multi-omics, including genetics, and develops innovative treatments using stem cells. In his laboratory, cutting-edge technologies are being created that bring Kazakhstan closer to the medicine of the future, surgery-free with a strong focus on early diagnosis and the regeneration of damaged tissues.